Hi guys, it's Easter Sunday. It's a very nice day, and what I wanted to do today is I wanted to talk a little bit about cell culture. Now, those people that know me know that I don't really like cell culture as a scientific discipline, and there are various reasons for that I will explain. What is cell culture? Cell culture is used in science a lot. You can take cells. Often the cells are cancer. Put together puzzle pieces, and that will help you understand how, for example, nutrients or chemicals may have particular anti-cancer effects. So if you grow a group of cells, you grow them on a plate or a dish, you expose them to, let's say, epicatechin from green tea, and you find that the cells have a reduced proliferation. Some of the cells are killed. In other words, when you treat them with the epicatechin, there aren't as many cells as if you treat them with just media. And from this, you can then do further studies whereby perhaps you look at particular enzymes within the cells, or you look at particular proteins to see how the epicatechin may be affecting the cells, and you can work out some molecular mechanisms. As a tool for scientists, that's great. There's nothing really inherently wrong with cell culture. In fact, it's actually an art. Those people that are very good at cell culture, not just the technical side, but also the theoretical side on how to select cells and how to and what to add, and experimental design and what type of chemicals you can add at what kind of concentrations. It's where science moves into an art, and those people are very skilled. And there've been some excellent cell culture papers, and some cell culture papers have taught us a lot about particular aspects of biology. The problem is that science is becoming a business, like everything, and it's going to the lowest common denominator. And the problem we have is that cell culture is very quick and easy to do, and you can publish a paper based on cell culture results quite easily. And this is really where the problem lies. Cell culture is being abused. It's being used by scientists really as a paper producing mechanism, so that they can keep their jobs and they can bring grants in, and they can push headlines in the press that aren't really justified by the data that they've collected. Now, if you understand how cell culture works and you're able to find whether particular headlines have come from cell culture experiments, I think it empowers you in terms of understanding nutrition. The purpose of my website really is to try and empower people to be able to understand nutrition themselves. I don't really want to have to tell you what to eat. Obviously, part of my job is to guide you there, but I don't really want to have to tell people what to eat. And I've always tried to say that really you're the ultimate arbiter of your own diet, and you really should make up your own decision, because everybody is slightly different. The only person that really knows what is healthy for you is, once you've educated yourself in nutrition, it is yourself. No one else can really do that for you. I don't make any money out of this, and really this is just a way of passing information to other people to help them empower themselves so they can make their own nutritional decisions. And I think if you understand cell culture and you understand the way that these papers are published, you can actually go a long way to dismissing a lot of the nutritional rhetoric that is out there in terms of the anti-cancer effects of particular chemicals. I'll give you a very basic example. If you take some cells and you add very high concentrations of something like ascorbic acid, which is vitamin C, you can actually change the pH of the media. Ascorbic acid is obviously an acid. It will go and dissociate in solution, and that will cause a pH change in the media. And that pH change can then kill the cells. Now, that's very interesting from a chemical point of view, and that experiment needed to be done at some point. But to say that that is an anti-cancer effect of vitamin C is a bit disingenuous, because really it's an anti-cancer effect of changing the pH of the media. Now, that's not to say that vitamin C doesn't have an anti-cancer effect, but that's not evidence to show that it does. You can do this with many different nutrients. Many different nutrients will act in very different ways in cell culture than they will in animal tissues. I'll give you another example. If you take polyphenols and you consume foods that are rich in polyphenols, they have antioxidant properties. Generally, when you consume foods like red wine and green tea, the polyphenols are very high concentrations, and that prevents autooxidation of the polyphenols in your gut, and they actually remain as antioxidants. And there is evidence that if you take in and you consume red wine, you consume green tea, these chemicals have an antioxidant effect in your gut. However, if you add them at lower concentrations into cell culture media, what you find is that they oxidize. In other words, they become pro-oxidants. What they were is originally they were antioxidants. Chemically, they change because of the cell culture media, and they then become pro-oxidants. And that pro-oxidant effect can actually kill cells. So it appears that they have a cytotoxic effect, and they do, but it's an artifact of the fact that you're using them at low concentrations in cell culture media in solution, and there's metal ions there, and that chemical soup there has changed them from their antioxidants, changed them from being antioxidants into being pro-oxidants. So you have to be very careful when you look at the results of cell culture studies, because you can very easily show that a nutrient has an anti-cancer effect when really it might not. And this is very important, because many of the headlines that come in the nutritional literature into the mainstream have come from cell culture. Very often you read such and such has anti-cancer effects, such and such may be an anti-cancer agent, and if you trace this information backwards to the original paper, what you generally stumble across is a cell culture study that's been done with some kind of cells, maybe human, maybe not human, maybe cancerous, maybe not cancerous. They've added the chemical, and they found that it's had an anti-proliferative effect. In other words, it's inhibited the growth of the cells. Well, that's great, but the headline often doesn't justify, is not often justified from the data. Sure, that is some evidence that you can use in a piece of a puzzle. You can put that piece with other pieces. Let's take green tea, for example, as an example, because I think green tea, of all of the nutrients that have been researched, green tea possibly has the most evidence from different parts of science. And I think if you put those pieces of evidence together, you find that green tea does have beneficial effects. Green tea certainly seems to have anti-cancer effects in cell culture studies. They've also done cell culture studies and in vitro studies where they've looked at the molecular mechanism whereby individual components within the green tea could affect cells and could kill cancer cells. There's epidemiological evidence, which is where they follow a large group of people, a large population, and then they measure associations with disease. There's evidence from epidemiological studies that those people that drink green tea have lower incidence of particular types of cancer, particularly esophageal cancer. There have been clinical trials that have been done whereby subjects have been fed green tea, and their antioxidant status has gone up. Various chemicals that may be markers of, for example, cancer have gone down, and so that is another piece of the puzzle. The biochemical response to drinking green tea has been measured, and the biochemical response to drinking green tea has also been measured in animals, and the animal and human study data is supportive of one another. Because animals don't live as long, you can actually do experiments where you induce cancer in animals, and obviously you can't do that in humans. And in animal studies where they've induced cancer through either the feeding of chemicals or the irradiation of animals, they found that green tea does decrease the rate of cancer formation. all these taken together, all these bits of evidence put together, suggest that green tea is protective of cancer. And in particular, green tea appears to be protective of esophageal cancer. And green tea may be protective of gut cancers in general because the green tea comes into contact with the gut cells, and that may be where it has its effect. So we also have to take this into consideration. Quite often cell culture studies will use cells that are body cells. They will expose chemicals to those body cells, and those, in reality, those chemicals may never actually, in the form they're tested in cell culture, they may never actually come into contact with those cells, because there's digestion, there's metabolism. When you digest, when you absorb nutrients, they're metabolized in the enterocytes. Some of them are passed back into the gut, where they're further metabolized by gut bacteria. Those that make it into the circulation go through the first pass metabolism in the liver, and they're further metabolized. So just because something is in a food doesn't mean it's going to get into the body, into the tissues. Very often things like flavonoids, all the polyphenols, are highly metabolized before they even make it into the body. We don't really understand what they're metabolized to. We've got a vague idea. We have measured these metabolites, but again, it's highly complex, so we don't really know what to test on the cells. So if you test, for example, quercetin on a cell culture in a cell culture experiment, you're assuming that the quercetin gets into the body. That might not be the case, because quercetin is glucuronidated in enterocytes. It then goes to the liver, where it may be methylated as well, and the chemical that you actually get in the plasma is not quercetin. It's a phase 2 metabolite of quercetin, and this is highly complex and is not really fully understood. So these experiments are very simplistic, and they can't be taken in isolation. This is what I'm trying to emphasize here. A single cell culture experiment, while interesting scientifically, is not nutritionally important. There have been some cell culture experiments that have changed the way that we viewed things, but that's usually when a new technique has come along, and when we've been able to measure something, perhaps a particular protein within the cells. Generally, a single cell culture experiment is not is not proof of anything, really. It's a piece of the puzzle that needs to be used in context. And unfortunately, what happens is, because science has become a business, because the media is very manipulative, and because there are agendas, people are selling things, people want to get this information out, people misuse the data, they misuse the results to create a headline that doesn't match the original data. And I think that this is one of the most important aspects of nutrition, where there is certain disingenuous information. There's a lot of misinformation that is passed out. I think many of the nutrients that are claimed to have anti-cancer effects, I think those effects are possibly questionable. That's not to say in the future that they won't be discovered, or there won't be more evidence added to them. But the data that's actually available it limits the scope of being able to suggest those kinds of conclusions. And if you search online for the amount of papers that have been done, have been investigated green tea in many different ways: animal studies, human studies, epidemiological studies, cell culture studies, in vitro studies, chemical analysis. Even green tea, the ability to say that it can prevent cancer is very controversial, and that is possibly the nutrient that has been researched the most extensively. So when you see the headline in the future, and you go on the internet and it says compound X is an anti-cancer compound, have a look, see if you can find the citation. If you can't, a quick search on Google Scholar or one of the similar search engines may bring up the paper. You can usually find it from the research team, and see if it's a cell culture experiment. If that cell culture experiment is performed in isolation, it's not considering other data, and there isn't really other types of research to go along with it. Really, you need to take that data with a pinch of salt. It's interesting. Read the paper by all means. Read the information, but don't be convinced that these compounds are beneficial unless there is good evidence to believe it. So I hope that was interesting. Cell culture is a bit of a minefield. It's a double-edged sword. Those people that can do cell culture well, and those people that use it as a tool, keep it in context, that's great. but it is abused, and unfortunately this is a reflection of the way science is headed. So I hope that was interesting. If you've got any comments on this, if you've got any questions that you want to ask me, please leave them in the comments box below this video. I will get back to you as always as soon as I can, and in the meantime, take care, and I will see you next week for another video.